All right, welcome back, everybody. We're going to be finishing up macromolecules today as far as our final unit for this chapter, as far as our final video is concerned. So let's find where we left off. Our last video, we were talking about proteins. We were discussing the different structures of proteins like primary, secondary, tertiary, quaternary. Just make sure you're aware primary is the most basic version of a protein, quaternary is the most complex version of a protein. Okay, all we got left to talk about are some special types of proteins. So first off, let's talk about enzymes. Enzymes are firstly, they're made up of proteins, so there's versions of proteins, as I just mentioned. The purpose of an enzyme is to speed up chemical reactions. So they accelerate reactions dramatically. Now, there are some special rules regarding enzymes. Number one, they're never used up, so they're never consumed. Number two, they don't use any energy. Enzymes don't require ATP, so that's really important. And number three, for an enzyme to be used, it has to be a reaction that can occur naturally. So a reaction that can occur on its own. The example I like to give is that a lot of the enzymes, like what you see on this slide here, are enzymes that are digestive enzymes. Digestion is something that can happen on its own, but with enzymes, that digestive process occurs much faster. So let's take a look here. I don't need us to worry too much about these enzymes yet. We're going to learn more about them in digestive system. But we have things like amylase, protease, lipase, catalase. If you notice, there's a bit of a common trend with the suffixes. ACE, that always tells you it's an enzyme. Easy. Amylase, just to go over them, helps to break down sugar. Protease sounds like protein, breaks down protein. Lipase sounds like lipid, helps to break down fats. Catalase is a unique one. It helps to break down hydrogen peroxide, which we'll learn more about in digestive. This is important. Enzyme action models. Kind of cut off the Y there, but enzyme action models. How does an enzyme actually react with the substrate, which is what you see as an S right here? The substrate is basically going to be the chemical that hasn't reacted yet, that still needs to react, or still needs to go through a reaction. So we have what's called lock and key and induced fit. Lock and key is by far the easiest process. For lock and key, the enzyme is going to bond to the substrate, and just like puzzle pieces, they're going to fuse together. That fusion together is going to cause the reaction to occur, and you're going to have the enzyme still, right? Because we never use the enzyme up. And we're going to have the products. The products are the end goal, the end result. The induced fit model is a little bit different. It's a little bit tricky. With induced fit, if you look at the enzyme and then you look at the substrate, you'll notice that they don't really fit really well. It's kind of like having two corner pieces on a puzzle. So instead, for the induced fit, I think of Play-Doh. We've all played with Play-Doh before. Play-Doh you can mold into any shape that you want. The enzyme itself will transition its shape. It will change its shape so that it can fit the substrate. And by doing so, you then have the reaction occur and you get the same result, products. So all in all, that's literally everything I just said here, but we're in out. So substrate and enzyme fit together perfectly, and induced fit is all about enzymes changing shape slightly to accommodate their substrate. I would make sure we are good with that. Factors that can affect enzymatic activity. This is a big thing. So these four factors we do need to know for testing purposes and just understanding macromolecules as a whole. First of all, temperature. Your body has an ideal temperature that you like to stay at. Now, because we use the imperial system in the United States, one of the few places that uses imperial instead of metric, the temperature is 98.6 plus or minus 1 degrees Fahrenheit. So that's your happy range. That's where your body is ideal. In the metric system, Celsius, it would be 37 plus or minus 1. Now, when it comes down to temperatures, our body is, again, sensitive to temperature changes. Your temperature changes all day, every day. You go out for a walk and it's hot outside, your temperature is going to climb. It's freezing cold outside, or even when you're sleeping, your temperatures will typically dip. But when you get to the extremes, so fevers or hypothermia, so really hot or really cold, your body has trouble handling that. And why that's such a problem is that it can actually affect directly enzymatic activity. As you add more heat, the enzymes activate more and more. Too hot means that the enzymes are reacting at a speed that's out of control. Too cold means that the enzymes are slowing down so substantially that they can't even do what they're supposed to do any longer. So there's a fine line, right? There's a fine balance. pH. As far as pH is concerned, pH is how we measure how acidic or basic things are, right? There's a pH for every single enzyme. We don't need to worry about that just yet. That's stuff that we'll learn more in digestive system. But as you notice, it depends on the part of the body where you're going to see pH differences vary. So the mouth, that's all about amylase, which is found in your saliva. That's about a pH of 7, which is neutral pH. That's the same thing as blood, same thing as water. Pepsin, that's an enzyme found in the stomach. Pepsin is going to be very important for digestive purposes, but pepsin has a pH of 2. Why is that? Because stomach acid is incredibly acidic. So this hormone or this enzyme has to be able to maintain itself throughout that. Trypsin is another really interesting one. We're going to learn about that in digestive, but that's found in the intestines. It's a little bit more alkaline, a little bit more basic. It's closer towards the 8 to 14 range. The concentration of enzyme and substrate play a major role as well. The way I can explain concentration is by thinking of it mathematically. I know people don't like to think about math, but this is the best way to approach it. If you have one enzyme and one substrate, when the reaction occurs, they're matched 1-to-1. That's a perfect balanced formula. For those of you that are taking chemistry that are watching this, balancing formulas is a big deal of chemistry. No problem. But what happens when you have two enzymes and one substrate? If you have two enzymes and one substrate, that's not enough substrate to maintain itself, so you burn through it too fast. Just like if you were to have half an enzyme and one substrate, that's not enough enzyme to work through the substrate. So at the end of the day, what number 3 on these factors is telling us is that you want to have the equal amounts of substrate and enzyme. That is the key. Last but not least, this is the easiest one, coenzymes. The term co as a prefix is to assist or help. So coenzymes are going to be things that help out your enzymes. And what helps out your enzymes? Minerals and vitamins. This is stuff that we consume from our diet. Okay? Don't have to worry too much about this, but this is more so proving a point. It's kind of saying, hey, macromolecules are found in every living being, every living organism, from a complex human to a single cell organism. We can see proteins like glycoproteins. Those are special structures found in the cell membrane that help to communicate with other cells. We can see literally the cell membrane made up of lipids and cholesterol, fats. Inside the cytoplasm, you're going to find a lot more carbohydrates. And then deep in the cell, you can't really see it in this picture, you'd find the nucleus. And what's inside the nucleus? DNA, right? Nucleic acid. And that's it. This video can be found on YouTube as well. If you look up in the YouTube search bar, Biological Molecules Crash Course, it'll pull you into the biology review. Unlike my other videos that I share crash course videos for anatomy, this one's a biology-centered one because this unit is literally reviewing bio. So that's kind of why. But it's a great video to watch if you just want to recap in like 12 minutes everything that we discussed in this particular unit. Okay, so I think that's a great place for us to finish up. Other than that, we're all set. Thanks, guys, for tuning in for this unit. I'll see you guys when we reach the next one, which should be integumentary system, all about the skin.